Hi, thank you for joining our webinar today. Today's topic is Introduction to Bioconjugation. This will be presented by my colleague, Dr. Craig Powell. He is the Director of Technical Services at Vector Labs. Today's webinar will be about Solulink Bioconjugation Chemistry and its applications. During the 1-hour presentation, we will have a quick FAQ session at the end. Please submit any questions you may have using the chat function to webinar site. I now introduce you, Dr. Craig Powell. Over to you, Craig.
Thanks very much, Byron. And hello to everyone joining us for today's webinar. You know, some listeners may be familiar with our previous webinars or webinar series that focused on applications such as immunohistochemistry and immunofluorescence. Now, as Byron mentioned, today's webinar is on the topic of bioconjugation, which is a little different from our previous webinars, though it's not unrelated, as you'll soon see once we get into the presentation. All right, so here's a quick overview of what we'll be discussing today. We'll look at what is bioconjugation. We'll look at a bit of a definition on that. We'll look at some common examples of bioconjugation, some of the nomenclature and phrases that are used. We'll dip our toe a little bit in some organic chemistry. Then we'll pivot over to look at some really interesting case studies on how bioconjugation is being used to push the frontiers of both medicine and science. Hopefully that might pique some interest for some of you. Then we'll look at the actual Solulink chemistry itself, the bioconjugation technology that's incorporated in those products. Then we'll have a look at what the product portfolio is, focusing on some of the kit formats, and I'll mention why we're focusing on that a little later on. And then tips and tricks or do's and don'ts for bioconjugation, some tools and resources that we can make available for you. And then, as Byron mentioned at the end, we'll have some time for a little bit of a Q&A. All right, so let's dig right into it here. What is bioconjugation? And the image I've got here on the left-hand side is probably the easiest one to talk from or visualize at your end. So I've got a molecule over here, a biomolecule that looks suspiciously like an antibody, and you want to conjugate this squiggly line to it. Let's just call this squiggly material peroxidase or horseradish peroxidase. And I'm going to use this example throughout the presentation because it'll be easier just to stick with the one example to try and convey some of these concepts about bioconjugation. So again, we've got our biomolecule, the antibody here. We want to attach peroxidase to it, and that's basically what we do using chemistry to generate a bioconjugate. And this is in fact then a peroxidase conjugated antibody. And this could be a primary or a secondary antibody, for example. So bioconjugation is the chemical linking of two molecules to form a single unit or a hybrid. But since this is bioconjugation, of course, at least one of those molecules in this union or hybrid, you know, would be a biomolecule such as an antibody, a peptide, a protein, or indeed an oligonucleotide. Now, one thing that's very important is the conjugate retains the activity of each of the components. In the example that I've got over here, the antibody, of course, you want that to still bind to its target antigen, and you want this enzyme, the example we're using here, peroxidase, to still be reactive with a subsequent chromogen or substrate that you might apply in a given application. However, another important point is when you do put these two together, you get an additional functionality that you don't get with either molecule alone. So this is the nuts and bolts of bioconjugation. And some of you might be thinking, hey, that sounds a little familiar. And so, yeah, indeed it is. Some of you are probably using bioconjugates in the lab on a daily basis. And on the left-hand side here, I've got an example of a fluorophore conjugated antibody. Indeed, this could be a primary antibody. It could be a secondary antibody, such as an anti-mouse, an anti-rabbit, something like that, where you would have a fluorophore. It could be a fluorescein, you know, your favorite dye on there, an Alexa, a dialite, whatever it might be. Indeed, you could swap out the word fluorophore for something like an enzyme, such as the example we just gave on the previous slide. Indeed, for a secondary antibody or even a primary, that could also be biotin or Doxogenic, and we'll be talking about some of those examples a little later on. So hopefully this sort of allays some fears for you about bioconjugation, what it is, and in fact you are probably using bioconjugates in the lab routinely or almost on a daily basis. Another example on the right-hand side I've got here is an agarose-bound lectin. This could be certainly an agarose-bound antibody as well, but these are agarose beads conjugated with, say, this glycoprotein or lectin, whatever it might be, and then applied in a column format for isolation or capture of whatever it might be. So that's another example of bioconjugation whereby that entity, whether it be an antibody or glycoprotein, is bound to that matrice, these beads in the example I've got here, and then utilized as a tool to extract or capture or isolate a given target antigen or protein, for example, of choice. All right, so now perhaps what I should put up here, or what I should have put up here, is a little warning flag saying organic chemistry ahead. So just bear with me over these next couple of slides and don't be too concerned about some of these structures. I'm going to walk you through these, but after these couple of slides, we'll get into some of the case studies. But I feel it's very important, particularly with an introduction to bioconjugation, to become familiar with some of the nomenclature or phrases that are used out there. Otherwise, if you're unclear on that, then you don't get the full appreciation of what we're talking about. So here are a couple of commonly used names or phrases when we're talking about bioconjugation. And one of them is a functional group. And this is part of a molecule that reacts with the reactive group to form a bond. And the example I've got here again is our antibody. And we've got an amine group here. And usually on antibodies, the idea is to target primary amine groups. There's a lot of them usually through lysines that are present. And it's very easy to use this functional group to then attach other molecules, or linkers I should say, to the antibody. And I've got an example over here. Here's the linker, this one over here. And those of you who may be familiar with some organic chemistry or perhaps have done some bioconjugation before may identify this molecule as biotin NHS. And basically, just to walk you through it, this is the biotin component here. This is the part that will be accessible for an avidin or a streptavidin subsequent detection conjugate, or indeed an anti-biotin antibody. And then the reactive group over here, this particular group, binds to this functional group linking chemistry. And this is part of the link. The reactive group is part of the link that reacts with this functional group on the molecule to form a bond. Now, so we've just discussed what a functional group is. That's part of the molecule. The reactive group reacts with that functional group, and it's part of the linker. Another part of the linker here is this spacer arm, which separates, or joins, I should say, these two ends of the linker. Now, spacer arms come in a lot of different flavors, I will say, and we'll touch on that a little later on. But basically, a spacer arm, as you can see, separates the reactive group from the business end, the biotin end in this case, of that linker to give it accessibility for subsequent binding by an antibiotin or a streptavidin or avidin molecule. All right, so just become familiar with some of these terms: functional group, reactive group, and spacer arm as we go through the presentation. All right, so here is another couple of terms you may encounter when you look at some of the options for bioconjugation. One here is termed a Homo bifunctional linker, this particular phrase up here. Another phrase is a heterobifunctional linker. And the example I've got for a Homo bifunctional linker is actually glutaraldehyde. And many of our listeners out there might be familiar with using aldehyde-based fixatives for tissue or cell staining for subsequent immunohistochemistry or immunofluorescence. And indeed, this is a great example of their action or function in that they do cross-link proteins. And if you look at glutaraldehyde here, it has on either end these actual aldehyde groups. And you can imagine if you do apply glutaraldehyde with an antibody, it would react with the amine groups. You would get an antibody binding here. And similarly, you get an antibody binding here. So these Homo bifunctional linkers have the same reactive group on either end of the molecule. You can imagine if an antibody was bound here and you had another glutaraldehyde, you get another antibody, and of course the linkage would go on. So Homo bifunctional linkers limit what you can do a little bit with the bioconjugate afterwards. And indeed, the example I've given here, you'd simply just get a big ball or polymer of antibody, which may be useful in some applications, but certainly not for more specific applications. So usually investigators use something called a heterobifunctional linker, which is different from a homo bifunctional linker in that it has different groups at either end of the linker. The example here is a sulfo NHS ester. This reacts with the amine functional groups. You can remember on a previous slide we were talking about a functional group on an antibody. So this would bind, this end here would bind to the antibody, and then it has a different end over here, this maleimide, that reacts with these thiol or sulfhydryl functional groups of a different molecule. So you can certainly then utilize this, and this is a molecule called sulfo SMCC. You can certainly look at that. It's a very popular molecule for conjugating to antibodies and other components as well. All right, so this is pretty much the extent of the biochemistry. We'll be talking about organic chemistry. Now we'll look though at some of the other selections or choices that are commonly used for binding or bioconjugating proteins. And we have used the term previously in some other slides, amine groups. We have looked at some sulfhydryl groups. And so here's the accessible group. This is the name of the particular group. And this is the actual target structure of it. And then it reacts usually with something like one of these. And we have mentioned NHS esters before. So whereby you'd have this amine group on an antibody, you'd react with, say, a biotin or say a fluorescein NHS ester, and that's the way you would then get a biotin or a fluorescein attaching directly to that antibody. Some of these other groups, sulfhydryl groups, aren't freely available on an antibody, and so the antibody itself would have to be modified, usually through a reducing agent, to cleave the antibody to make these sulfhydryl groups available for subsequent reactivity with the maleimide group. And that maleimide indeed may then be conjugated with a biotin or a fluorescein itself, but the antibody itself would have to be modified. And the same thing with some of these other particular groups like aldehydes and azides are not present on antibodies, so the antibody itself would have to be modified in order to use a given linkage or reactivity or bioconjugate methodology. And so certainly this is just an abbreviated list of some of the many ways you can attach linkers to proteins, for example. And I'd say the most commonly used ones are the amines that's listed up here, as I mentioned, very prominent and available and accessible. That's another key word. While something might be present on an antibody or indeed a target that you want to bioconjugate, the quick question is, is it accessible, or do you have to modify it somehow to get access to that functional group so you can then put a linker on it? So since amines, primary amine groups, are readily accessible on things such as antibodies, they're usually the most popular method of targeting or conjugating antibodies. And then I'd say the next is probably the use of these sulfhydryl groups through some modification. We'll be talking a little bit about the linkage of an aldehyde and hydrazine, which is the basis of the Solulink chemistry, a little later on. All right, so some of you might be thinking, hey, this is great, Craig. So, I'm getting pretty interested here in bioconjugation. I've always wanted to know about it. So what are some of the considerations if I want to launch into a project? Well, you want to step back a little bit and have a consideration about, well, what is the actual intended application and how is it going to be performed? Is this going to be performed in solution, for example, or indeed is it going to be a bioconjugation on a hard surface or a matrice, a bead, such as we mentioned before, an agarose bead? And what is that biomolecule that you're going to be conjugating? And indeed, the 2 points there, what is the intended application and what is the biomolecule, will probably lead you then into the choice of chemistry. You know, what is available for conjugating or accessible, what reactive group or functional groups, I should say, are available on that biomolecule. And once you've sort of looked at those options, a further one would be a spacer arm selection. If you can remember back to the diagram I showed earlier on of that Biotin NHS ester, we had that spacer arm that essentially joins the reactive group with, you know, the biotin of that example. And the spacer arm can vary in a number of different ways. I mentioned it comes in different flavors. And what I meant by that is it comes in different lengths and different composition, which may affect things such as solubility. And also, there is another factor there that could be important, and we'll touch on this a little later on, is whether or not you can actually cleave it. And what that means is by splitting it using something such as a reducing agent, or indeed, as we'll see later on, a UV light, for example. And that may add functionality to that bioconjugate. Certainly solubility and stability. You know, are you working with a labile molecule that might not be soluble in, you know, aqueous phases, or, you know, does it precipitate out in organic solvents like a DMF or DMSO, for example? And also then stability in solution prior to the conjugation, and also stability after conjugation. Does it have a very short half-life? Does it, as I mentioned just then, crash out of solution? Can you actually use that in a functional way? And then consideration, once you've done the bioconjugation, you know, think about what you're going to do with it for subsequent, you know, detection or capture visualization methodologies, isolation and things like that. So you can see this is, again, it's just an abbreviated list of considerations when you're starting to get into bioconjugation. It can get complicated quickly. And indeed, you know, the title of this particular webinar is Introduction to Bioconjugation. And so you could certainly go off right now and we could wave you off and say good luck with using separate linkers like the Biotin NHS ester. But I think you'll find pretty quickly that there are a number of other considerations that you'd have to take under consideration, such as, you know, pH, you know, buffers, salts, things like that. How are you going to purify it as well? That may not be readily available. It may be expensive to do or a little complicated. So we're going to have a look at some of the kit formats coming up here that I think would be a great way to introduce yourself, or indeed a lab member or your lab, to bioconjugation. But let's just take a step back and have a look at some of the applications where bioconjugation can come in. And it doesn't sort of happen in a vacuum. No one sort of wakes up and goes, hey, I'm going to conjugate an antibody to fluorescein today. Usually, and I'll walk you through this slide, usually it starts with a research project. Over here on the left-hand side, you're thinking, you know what, we've got whatever it might be. You know, you're looking at something you want to capture or indeed isolate a given protein. You want to detect it or visualize it. Maybe you're trying to upregulate something and you want to quantify it. I've got the word there, perturbation. Maybe you want to interfere with a cellular cascade or pathway, and you want to inhibit something or stop it and see what happens. And that's just an abbreviated list then again. And you're thinking, well, how am I going to probe this? How am I going to you know, undertake this particular research project? What you do then is think of various investigative sort of techniques in doing that, whether it be using flow cytometry, a fluorescently conjugated antibody, some cell or tissue-based staining, microscopy such as electron microscopy, for example, or immunoassays such as lateral flow, protein or Western blotting. Maybe you'd quantify something with an ELISA. Surface plasmon resonance is certainly gaining popularity out there. Maybe you want more defined drug delivery, and I'll show you an example of that coming up. Or indeed, maybe you're working with an omics platform. And what I mean by that is whether it's proteomics or a genomics platform. And indeed, when thinking about that, and indeed all of these investigative sort of techniques do incorporate the use of bioconjugates. And it might be that, you know, your favorite vendor, for example, whoever that is, you know, doesn't have all of the repertoire of conjugates, you know, that would help you probe or generate the results you want in a given research project. Maybe you could take it further by having, you know, an antibody conjugated to a novel enzyme or a fluorophore, or you need to plate it somewhere to capture or isolate material. And the same thing for some of these other particular biomolecules as well. Hopefully the idea of this slide is to start getting the juices running in your mind and thinking, okay, how can I push my research project further? Or what if I could quantify, you know, perturb or whatever it might be, you know, a given cascade or signaling pathway in my study? Could you push that particular research further and probe new areas? And this is where the bioconjugate tools come in. Okay, so we've just gone over what is bioconjugation, some of the common examples and the nomenclature. As I mentioned, we just dipped our toe a bit in some of the organic chemistry and looking at some of the applications here. Hopefully it's starting to get you a little interested. Let me show you a couple of case studies where people are using bioconjugation, and while I'll say where bioconjugation is vitally important in generating the results they want. The first case study I've got here is using an antibody drug conjugate. And essentially some investigators conjugated a humanized antibody to a given drug. And let me work from this particular image on the right-hand side. So we've got a human T cell here that expresses this particular antigen, CXCR4. The investigators were trying to get this particular drug that's made by our friends at Bristol Myers Squibb to be a little more selective in the way it does target its particular antigen and uptake in that given cell type. So it's not particularly specific, the drug. So the investigators were thinking, well, you know what? If we target this particular expressed antigen on these human T cells with an antibody, we conjugate the antibody, and they did that using Solulink conjugation chemistry to this drug. It can then get incorporated into the cell and work on these particular tyrosine kinase pathways and affect T cell activation. So hopefully that makes sense. So the drug itself actually is a tyrosine kinase inhibitor. And so by using the specificity of that antibody to bind to that target antigen, and they did studies to identify that once it binds to that antigen, it actually does get incorporated inside the cell and therefore acts specifically. And because it has that drug payload, there it can actually act in situ in that given cell. And so I thought that was a pretty neat way of looking at bioconjugation. And I'd call it, actually I did read somewhere, someone called it a vectorized, or I'll call it targeted chemotherapy, rather than a whole systemic chemotherapy. This is directed toward a given cell type and antigen. So I think this is pretty neat. And there are now, I think, last count, around about 80 or so antibody drug candidates using various, bioconjugation methodology under evaluation by the FDA. So pretty neat stuff that's going on there with bioconjugation. Here's another one. It's where they use bioconjugation with the Solulink technology, DNA antibody bar-coded arrays. These particular investigators developed this platform called a MIST platform, a multiplex in situ tagging platform, whereby they had a monolayer of single-stranded DNA microbeads on, I think this was like a glass surface, if I remember correctly. But these are all immobilized. They're in the one place, and they've got a different single strand on them. That's the idea of the colors there. But by using bioconjugation, what they did was attach complementary DNA to antibodies that then hybridized to those single strands, which allowed these antibodies specifically to capture different proteins in solution. And then using a fluorescent sandwich ELISA, they could quantify that. Later on in the study, if you're going to look at it, they actually then eluted off those complementary DNA strands, and they could do several rounds or multiple rounds of detection. So this allowed them to do high throughput with high sensitivity. But it was this conjugation of the antibody with that complementary DNA strand that allowed them to undertake this given methodology. Okay, one more case study we'll look at that might be of interest to investigators out there. This was actually just came out last year, and these investigators really wanted to develop a straightforward method to probe or investigate tumor heterogeneity without going the way of what I will refer to as maybe a complicated, complex, and indeed a high-cost platform that I know are out there. And what these investigators did was they developed what they referred to as a cyclic or a cyclic immunofluorescence methodology. Basically, they conjugated 14 different antibodies to oligonucleotides, and they put these particular oligonucleotides down on the tissue at the one time. So there was not subsequent incubations of separate different antibodies. It was 14 antibodies with defined oligonucleotides, unique oligonucleotides on the tissue at the one time. And then they put on a complementary fluorophore oligo strand and did some visualization and identification with that. Because it also had a photocleavable linker, if you remember back, we did talk about spacer arms and how they could have an option of having a cleavable arm. This one was a photocleavable arm or linker arm that they could break off using UV light exposure, and then for that allowed them to do reprobing later on with some other targets. So this actually is a very neat way for those of you out there who may be doing multiplex immunofluorescence or things like that. Some of you might be thinking, hey, there's no secondary antibodies here. And that's exactly right. These investigators were not hobbled, if you will, or hamstrung by having to use defined anti-mouse or anti-rabbit or anti-goat secondary antibodies and constrained with those parameters. They utilized these antibody oligo conjugates and these unique complementary strands, and they didn't have to worry about secondary antibodies. So I think that was pretty neat, and this is certainly going to be something that's developed forward, and I do believe that other platforms are very similar out there at the moment. So hopefully this gives you some idea about how bioconjugation is being used out there, and it's certainly coming to prominence in a lot of methodologies. Okay, so we've just had a look at some interesting case studies. Let's have a dive now into the Solulink chemistry. And the question I've got here, what is Solulink bioconjugation? And let me walk you through this slide. It's really a couple of different steps, but it involves the use of this molecule called HYNEC and another molecule called 4FB. And obviously there are abbreviations for much longer chemical names. But nonetheless, let's break it down a bit, and we'll use the example we've been using since pretty much the first slide here in the webinar. Here's our antibody, and we're going to conjugate this enzyme horseradish peroxidase to it. So for the Solulink conjugation, first off, you attach HYNEC to the antibody, and in a separate step, you attach 4FB to the enzyme. So they're two separate steps, and you do those number one. And once they're done, once they're performed, then you mix them together And given the high affinity of the high nick for the 4FB, that linkage, you get essentially this bioconjugate. Peroxidase anti-mouse is, I just made that up. It could be anything you want. It could be a primary antibody, a secondary antibody, but the idea being that you've got a peroxidase conjugated antibody. And the nuts and bolts, the heart of the Solulink bioconjugation chemistry is the use of this high nick and 4FB molecule to link together and form these bioconjugates. All right, just take a deep breath, another couple of chemical structures here, but I think this is important just to get into it a little bit more. Here's the high nick molecule right here. This particular end reactive group will bind to the amines on the antibody, and similarly on the 4FB that will bind target amine groups on this particular enzyme, the peroxidase. And some of you might be looking at this 4FB molecule and just thinking, hey, didn't we see that on the glutaraldehyde? And the answer is yes, you did. So this is actually an aromatic aldehyde that links to an aromatic hydrazine, and with under certain conditions, once they're bound to their given targets there, they give you this particular bioconjugate. And interestingly, you'll see right in the middle here is this hydrazone chromophore. And the idea here of the graph is to show you you can actually use that or utilize that linkage, that chromophore in the linkage, to identify that, yes, it actually has been conjugated and how much is there. All right, so we're using this particular example as again throughout the webinar just to get some of these concepts across. So again, here's our antibody, here's the peroxidase enzyme, here's the linker, and this particular linker using high nick 4FB has this hydrazone bond that enables quantification of that actual linkage to occur. And so that's one of the key benefits or advantages of using the Solulink chemistry, is that you can quantify how much material is bound to your biomolecule. And I've listed here some of the other features or advantages of the Solulink chemistry. Certainly, the reactions occur under relatively mild conditions, and that's very important. What I mean by that is it's not at extremes of pH, not high alkalinity or very low pH, because certainly if you are working, I used the word labile before, with some, or delicate, I'll use that phrase, maybe that's a little better, some delicate specimens, you want to retain maximum activity or binding activity. And certainly for antibodies, you want them to bind to their target antigen. You don't want to break them up or things like that. So that's very important. Some other linking technologies can be rather harsh, and you do lose activity. So just be aware of that. Some of the binding gets very close to 100% in terms of its labeling efficiency. If you selected or chose a linking methodology that only put a linkage on 10% of your biomolecule, you're obviously losing, I should say, about 90% of your biomolecule, and that's going to flush down the sink, and you'll lose a lot of that. So you really want a high efficiency labeling methodology, which this is. You also want it to work relatively quickly. You don't want to be sitting around for hours to have the linkage occur. So indeed, Solulink chemistry incorporates a catalyst called aniline, which drives the reaction very quickly to form that conjugation. Stability too is a key factor, and I hinted on that earlier on under the considerations for bioconjugation. You want certainly the reagents and the conjugates to be stable once formed. And certainly, a year in storage at 4 degrees is a very good time for a bioconjugate. Consistency between lots is pretty crucial, particularly if you're trying to duplicate studies. Scalability, if you're going to amp it up instead of just a very small, maybe milligram amounts, you want to go to or microgram amounts rather, you want to go to milligrams or even gram quantities. Certainly that's a methodology you can consider or an approach. And we did mention quantifiable, but also it's very versatile and flexible. It can be used in many applications. And if you think back to the slide we mentioned about the bioconjugate applications with the image of the cell there, all of those particular methodologies we listed, flow cytometry, microscopy, etc. You know, this particular bioconjugation methodology can be used in those studies. All right, let's have a dig now into the Solulink product portfolio. And I did mention earlier on, we're going to be looking at the kits themselves, or focusing on the kits. And I've split them into two groups. We'll look at the antibody and protein conjugation kits, and we'll look at oligonucleotide conjugation kits. So here's the selection of antibody conjugation kits, and the reason why, again, I'm focusing on the kits, just to reiterate or emphasize the fact in that, again, this is an introduction to bioconjugation, and I think a very easy way to become familiar with the techniques, the requirements, would be to go with complete detection kits, something that includes all the necessary components and reagents to perform the bioconjugation reliably, to give you confidence then to go forward and perhaps use separate linkers later on or explore other opportunities. So that's why I'm focusing on the kit formats. These are complete detection kits, or complete conjugation kits, I should say rather, that include all the necessary components. A great segue into exploring the really exciting field of bioconjugation. So there is a Biotin endodoxagenin kits. We've got a phycoerythrin kit, fluorescein labeling kit, and perhaps not surprisingly, a peroxidase conjugation kit as well. So very quickly, we have two biotinylation kits. One is specifically for antibodies. It's referred to as a one-shot antibody biotinylation kit, and another one would be for proteins other than antibodies, and I've listed it here. And Biotin's used for a lot of different reasons, and I'm sure that most of our listeners out there have used Biotin conjugates in one or another different formats. So both of these biotinylation kits, referred to as Chromalink Biotin kits, use this particular compound, and this is similar to the Biotin NHS that I showed earlier on. If you look at it, you'll notice very quickly that these Biotin compounds are indeed heterobifunctional linkers. Here's the reactive group that'll bind to your primary amine groups on your antibody, and here's the Biotin over here that will be accessible for subsequent avidin, streptavidin, or indeed an anti-Biotin antibody. What is different here, though, is there is a very long chain spacer arm. It's a hydrophilic PEG spacer arm that helps in solubility of the particular linker, and also it separates the Biotin a little further away from the antibody to prevent things such as steric hindrance, to enable this Biotin to be accessible for subsequent binding. And you will see here, as we've been mentioning about the conjugation of the HiNic and the 4FB, you do get this hydrazone chromophore being incorporated in this linker. And that's important for a couple of reasons, particularly with these biotinylation kits. Number one, you'll see here that there is a significant time saving in that you don't have to run a Haber assay. A Haber assay is a colorimetric assay. And I've got here that some other kits do include that or recommend running a Haber assay for Biotin quantitation. But with the hydrazone chromophore, you can get away with just a 5-minute UV scan to identify that conjugation has occurred, and you can actually quantify the Biotin. But the other thing that my colleague Byron always reminds me of is that the Haber assay underreports the amount of, I'd say consistently underreports, the amount of Biotin that's actually incorporated on a given target. So in that case, it's an inferior way of measuring or quantifying Biotin in these particular bioconjugation methodologies. And certainly this just shows you how you can use this particular chromophore. You read it at 354 nanometers, and this is actually a bovine IgG biotinylated with one of the Chromalink Biotin kits. This shows you, biotin to protein molar equivalents. Obviously, when you read it under the spectrophotometer at that given wavelength, you can easily quantify the amount of biotin that's on there. Okay, this brings us to the Doxogenic kit. Very quickly, here's the dig over here that'll be bound by an anti-dig antibody. Incorporated here is this long chain hydrophilic peg spacer, performing very similar, solubility roles that I mentioned earlier for the biotin. Here's this particular chromophore again to allow or enable quantification with that quick UV scan. And here's the reactive group that'll bind to your amine groups on the antibody. So this particular kit, the Doxogenic antibody labeling kit, allows you to link the Doxogenic onto an antibody, for example. You're thinking, okay, that's great, Craig. What sort of applications can I use these for? And I've just listed over here on the right-hand side, just some bullet points on how they can be applied. One of the more interesting applications here actually was multiplexed immunofluorescence staining, whereby two mouse primary antibodies, I should say one was conjugated with the biotin, another one with the Doxogenic kit, and then subsequently detected using other fluorophore conjugates. But nonetheless, I've got an example here of a sandwich ELISA using a Chromalink Biotin kit. This particular capture antibody was plated, the antigen was captured, and then this detection antibody was conjugated with Chromalink. Here's your streptavidin, your enzyme, and then they read that particular reaction. So this just gives you some ideas of how these kits are used out there. We also have the point here about using magnetic beads as well. Could be a streptavidin magnetic bead for pulling out biotinylated antibodies or proteins, for example. Right, so let's toggle now to just some of these other complete antibody kits that we've got. And one is the phycoerythrin kit. It puts a red fluorophore phycoerythrin on antibodies. And we've got the peroxidase antibody kit as well. And this particular diagram here just shows what we've been representing all through the webinar. Your antibody being bound by the HiNic compound, the HRP enzyme conjugated to 4FB, and then under the certain conditions and using the aniline catalyst, driving that reaction to get this linkage and the hydrazone bond that you can then quantify. The particular one kit that there's a bit of a standout here is the Flourishing Antibody Kit, and I've got a couple of key points there about it. It actually does not use HiNic and 4FB, so it's a bit of an outlier. It actually uses an NHS fluorescein to perform the conjugation. But nonetheless, this particular kit, it is a complete kit that enables labeling and purification within about 90 minutes. So that might be of interest to some investigators out there. Some of the other applications that these kits would be used for, here are these fluorophore antibody conjugation kits. Probably flow cytometry would be a very popular methodology. Can certainly be used for blood cell ELISAs, various immunoassays, and the peroxidase antibody conjugate kit, certainly for very similar applications as well. Immunoassays could be something such as an ELISpot, I've got there, or maybe a lateral flow assay. One further kit I'd like to draw your attention to is a protein-protein kit, whereby, let's just say, I don't know, maybe you want to conjugate a hormone with a fluorophore or an enzyme or something like that, or maybe just BSA, for example, or albumin as a control with whatever it might be. Certainly this particular kit does incorporate the use of HiNic and 4FB as well, and enables that hydrazone linkage to occur. And some examples of applications would be in vitro assays. And I do have an example here of an antibody, whether it be a primary or secondary to a novel enzyme or a fluorophore, but also in vivo assays. And we did touch a little bit on these ADCs or antibody drug conjugates in one of those case studies earlier on. Indeed, it could be an antibody digest. There's a lot of work going on with antibodies, an FAB, FAB2 or something like that, to various drugs or whatever it might be. So hopefully this is giving you some ideas about how bioconjugation could potentially be looked at through a different lens. Let me put it that way. All right, this brings us to the Oligo Conjugation Kits, and there's two choices. One is a protein oligonucleotide kit, and the other one is an antibody oligonucleotide conjugation kit. And both of these use, again, the Heineck 4FB linkage technology. You do get this hydrazone bond that allows you for quantification of the conjugation to occur. And examples I've got here of where they might be used, and some of you may even be using these applications or techniques out there, proximal ligation assays, whereby you might have different antibodies labeled with different oligos, and when they're in close proximity, obviously you get that hybridization occurring. Similar would be an immuno-PCR reaction, and also targeted oligo delivery. And I would say that that's probably something that's coming to the forefront. And what do I mean by targeted oligo delivery would be, maybe the use of something like an siRNA or like a small interfering RNA, which really acts, I'd say, through enzymatic breakdown of complementary mRNA, whereby you can then interfere or perturb, there's that word again, various, progression of cancer or something like that. And, there's been, I'd say, a lot of work being done about how to extend the half-life of siRNA to be more of a targeted approach. So there's some interesting work going on about that, but that's where one of these oligonucleotide, conjugation kits would be of interest. All right. Tips and tricks, or I've got here in the table do's and don'ts. So really quickly, do determine the antibody volume and concentration, and don't automatically assume that what's provided to you by a vendor is actually the correct volume or indeed concentration. The vendor might be overfilling the material by a 5 or 10%, so that you're guaranteed of getting whatever it is concentration they say you're getting. So make sure that you do then validate that, make sure you adjust the volume and concentration as required, and work within the given parameters as per the kit instructions. Don't use too little, don't use too much. Get into that sweet spot of volume and concentration. Do remove preservatives, and what I mean by that is things such as azides or antimicrobial agents that may interfere with the conjugation. And don't automatically assume that, oh, my antibody's purified, I can continue straight away with the bioconjugation. Some of these purified antibodies may be supplied in buffer with carrier proteins such as a BSA, or indeed I've got here other amine molecules. And what I mean by that is contaminants or indeed buffers, like a tris-based buffer is how the antibody is supplied, which tris can be actually conjugated. So through the use of NHS esters or something like that. So just get rid of those before you continue with the bioconjugation. Certainly, if you are using oligonucleotides or peptides, ensure the appropriate purity that's been run through HPLC, for example, and modified. So, for example, don't use, I would call it dirty material, something from a serum bleed or something like that. And certainly for the Heineck and 4FB conjugation methodology, make sure they are amine modified so that you can link the 4FB onto those, or the Heineck onto those given molecules. And at the end, I would highly recommend removing unincorporated label. And don't leave it around. That can cause a lot of problems with perhaps interference with binding subsequently, or high background or something like that. So do remove the unincorporated label. All right, this brings us to the end here. We do have some tools and resources available on our website. You can access these through this particular linkage. We've got a white paper here talking a little bit more in depth about the Solulink technology. And we've recently made available a bioconjugation resource guide that highlights the products and kits I've just spoken about, but also some of the separate linkers and magnetic beads that are available as well. Some of the other tools and resources available on our website, we've got product manuals, online calculators I've listed here that you can, that I should say would make the process a little easier for bioconjugation. Troubleshooting guides, you can go through and make sure that everything is looking as you would expect. We're building out an FAQ section, and we do have some published references available as well. Okay, so that brings me to the end of the presentation, and with that in mind, now with the information I've bestowed upon you, go forth and conjugate with confidence, but I'll open the floor up now to any questions you may have. Thank you.
Thank you, Craig, for that great presentation. We will proceed with the Q&A. The first question is, if I am using expensive material, antibodies or oligos, how do I maximize conjugate yield with the Cyclic and 4FB chemistry?
Yeah, certainly. Number one, as I was just talking about some of the do's and don'ts there, you know, what you want to do is use a highly purified material. You know, certainly, as I mentioned, for oligos, pass it through HPLC, or indeed you'd want to desalt it as well. And in some of these complete kits, there are things called Zebra columns that get it into a buffer format that make it more appropriate for bioconjugation. So you want to get rid of the contaminants, the preservatives, do a buffer exchange to get it into a good buffer initially to perform that bioconjugation. Make sure also that these particular biomolecules are within the appropriate concentration range. Certainly if you use too little, you might not get appropriate labeling. If you use too much, that's going to be problematic as well. There might be issues with solubility. It may crash out a solution. Another point would be volume as well. Don't use very small amounts, because otherwise you just might lose the material when you go through a purification step at the end, trying to remove the unincorporated, or indeed using too much material. You've just got everything slopping around there, and you might reduce the labeling efficiency there. You know, one other point I didn't really touch on in the presentation was that with the Cyclic and 4FB linkages, there are actually what we refer to as checkpoints during the bioconjugation methodology. And what you do is you check the MSR, the molar substitution ratio, which is really, you check how much Cyclic is attached to, for example, the antibody, and you check how much 4FB is on your, for example, enzyme, that same example I've been using, and make sure you've got the appropriate, you know, molar substitution ratios of those molecules on those given entities before you continue with the bioconjugation. Because that's the great thing about the Solulink chemistry, they're midpoint checks to make sure that, yeah, okay, I've got, you know, my estimated, you know, 3 to 5 4FB on my enzyme and 3 to 5 Cyclic on my antibody. You know, all signs point to a successful conjugation, and therefore you move forward, and then you can use that hydrazone bond or chromophore to just again confirm the conjugation has occurred, and you can quantify that amount. So they're the sort of points I'd look at if you're working with expensive material or maybe precious material.
Great, thank you. The next question is, what is the difference between the protein oligo conjugation kit and the antibody oligo kit?
Yeah, I didn't really expand on that in the presentation. So as you saw for both the oligonucleotide kit, we've got an antibody and a protein oligonucleotide kit, and also for the biotinylation kits, we've got an antibody and a protein biotinylation kits. And that does pose the question, okay, if I'm not using an antibody, I can use the other, you know, another protein for the other kit. And the thing is, they are different a little bit in the way they are set up, in that the antibody kits have what I would say more of a highly stringent sort of step in terms of the purification, because we know that antibodies are, you know, around 150 to 160 kD in size, and therefore we include in those kits appropriate cleanup steps to make sure the material at the other end is highly pure. So with protein, Oligo Kit or the protein biotinylation kit, it's a little difficult to identify what given protein you might be using. It could be much bigger than an antibody, or it could be smaller. And so therefore it's a little more difficult to include in there a given cleanup step for that. So it gives you a little bit of an insight into those differences, or the differences, I should say, between those two kit formats.
Okay, great. The next question is, why should I choose Solulink over other bioconjugation kits such as Lightning Link?
Yeah, fair enough. I didn't touch on other technologies that are out there in terms of bioconjugation. I did show that there are other ways to perform protein conjugation with that abbreviated table. We looked at sulfhydryls and things like that. But I will say that the Lightning Link technology certainly does highlight how quickly it can be done. Interestingly, though, there's no cleanup step at the end. They put in a quenching reagent. But as I pointed out there, I think, if you're working particularly with precious material, or you're trying to maximize labeling efficiency, you really need highly reproducible results. I'd highly recommend performing a cleanup step, such as what we recommend in some of the methodologies for the Solulink chemistry. But again, what I've stressed throughout the presentation here for the Solulink chemistry is the use of the high Nickel 4FB, and the fact that they enable, one, as I just spoke about in an earlier question, this mid-checkpoint molar substitution ratio check, but also that hydrazone chromophore that allows you to do a quick UV scan to quantify that, yes, the conjugation is successful, and yes, I do have a known amount of whatever it is, biotin, for example, on my given protein or antibody. And that, I think, is invaluable for investigators who want that consistency, that reliability, that reproducibility between manufactured lots that they're doing in their lab. Otherwise, it's a little bit of a crapshoot with some of the Lightning Link stuff, and particularly if you're not using highly purified material, there could be a lot of problems with background and inconsistency in labeling, poor labeling efficiency, and things like that. So that's why we incorporate things such as these zebra columns for cleanup as well. Also, we'll say that we do have some online calculators and other tools and resources that I think would be helpful for investigators to look at as well.
Okay, great. The next question is about siRNA antibody conjugates. Can you elaborate on this application, and how do you remove excess siRNA?
Yeah, fair enough. So I did touch on that a little bit, and that was with the oligonucleotide kit. I spoke about targeted oligonucleotide delivery, whereby you'd use one of those kits. Maybe you'd have a protein, for example, and basically what some investigators are looking at with the siRNA is trying to increase its longevity. Once injected, I think it gets cleared pretty quickly by either kidneys or liver or something like that. And the idea is to apply it, conjugate it to a given target protein, so it can then be incorporated into a cell or cells, the target cells, and then it can do its business in terms of breaking down, complementary RNA. So introducing them has been an issue, and their longevity in this, as in when delivered systemically. So I think investigators are looking at things called protein cages. I think you can probably look that up in some of the methodologies that are there. And certainly I think those protein cages act to protect that small interfering RNA from getting gobbled up by the body and allow significant or substantial amounts then, or or I'll just say quantities to get incorporated into the cell at the place of entry. So, so once you do that, just certainly follow through the same methodologies that you would as per the the protein oligonucleotide kit in terms of cleaning that up as per those instructions.
All right, thank you, Craig. Our time is up for the Q&A session. Thank you for attending the webinar. We will send you the entire recording tomorrow through email. And if we were not able to answer your question, we will personally answer your question at a later time. And have a great day. Thanks for attending the webinar.
Thank you.